Okay, our next speaker is Barbara Wilhelm. She's from the School of Occupational and Public Health in the University of Guelph in Canada.
Close enough, okay.
Sorry. She's going to talk about a systematic review and meta-analysis of control methods for non-typhoidal Salmonella pre-harvest in cattle.
Thank you very much. Well, I would indeed like to tell you about our review of Salmonella control methods in on-farm cattle. I guess it should be obvious from the location, but I want to make clear immediately that in contrast with the presentation here this morning, I think by Dr. Smith, our interest in this question was from a public health perspective, not a clinical disease perspective. Okay. That works better. Non-typhoidal Salmonella is a very well-known cause of human foodborne illness, and recently there have been several large outbreaks of multi-drug resistant Salmonella associated with the consumption of beef products. This review was initiated specifically to provide data for an expert committee that was meeting to provide recommendations for Codex guidelines for control of Salmonella in beef and pork. So that's a huge topic, of course, and so we subdivided our findings by species, meaning pigs or pork versus beef or cattle, and also by point and chain. So this is an overview of what we did. We did a broad literature search in two databases. We did a search verification, which involved hand searching the references of randomly selected papers that we'd captured. And we also did a gray literature search to capture data from sources such as conference proceedings. And ultimately, we started with about 6,400 abstracts. And to each of those, we administered a questionnaire which basically asked, does the research in this abstract tackle some part of our large overarching research question? And if the answer were yes, we got the full paper, and then administered another questionnaire which confirmed relevance and also categorized by species, by point and chain that the intervention was to be administered, and location of sampling. And so ultimately we extracted data from about 540 papers. And you can see immediately that the majority of papers we captured looked at interventions in pigs and pork, so immediately I'm going to set all those off to the side. And you can also see that of the cattle papers, the majority studied interventions at processing, for example, use of acid washes and that kind of thing. A small number of papers looked at interventions applied at transport and lairage pre-slaughter. And so again, I'm going to set all those papers off to the side, and what I'm going to be talking about for the rest of this presentation is the 79 papers we captured looking at on-farm interventions. And so to try and bring some kind of order to this large number of papers, we subdivided into large intervention categories, and we designated them biosecurity, feed management, manipulation of gut microbiota, which is quite a mouthful, and since in cattle that consisted exclusively of probiotics such as lactobacillus, I'm going to just call it probiotics for the rest of the presentation, vaccination, and also other interventions. And so each of these large intervention areas, you can see, was underpinned by a variety of different study designs. And biosecurity, I guess not surprisingly, was studied largely by observational studies and also by a small number of stochastic or mathematical models. And in contrast, vaccination, which was the most studied intervention in our cattle data set, was investigated by controlled trials, where the investigators controlled allocation of the intervention, challenge trials where the investigators also controlled the infection, so it was an artificial infection. There were no quasi-experiments or before and after trials for the vaccination data set, but observational studies and again, mathematical models. And so looking first at biosecurity, and again, these were investigated largely by observational studies. The closed herd, although it was defined differently by different investigators, was maintaining a closed herd was consistently associated with reduced odds of Salmonella culture on farm. And several management procedures around calving time, two were predictors. If a farm fed colostrum only from the dam or the mother of that calf, and or used a dedicated calving area on farm, both of those practices were significantly associated with reduced odds of Salmonella culture on farm. Not surprisingly, manure management practices also were predictors. And so both use of flush systems in dairy barns for removing manure from the alleys, and broadcasting liquid manure relative to solid manure. Both of those were consistently associated with increased odds of Salmonella on farm. And the last one, hygiene, which was essentially cleaning and disinfection, that was really tough for us to summarize. It seemed like no two investigators defined it the same way, and possibly partly for that reason, we found inconsistent, and by that I mean not just variations in the magnitude of association, but changes in the direction of the association of hygiene with culture across different studies. So when we looked at the probiotic data set, you can see that we were able to pool findings from 4 trials that were reported by 2 different investigations, all looking at the effects of feeding probiotics to feedlot cattle, measuring the outcome of fecal shedding. And so we estimated a borderline significant odds ratio. And the grade rating I'll just touch on briefly, although I'd be happy to talk about it after. It reflects the fact that we were directed for any data set for which we could do meta-analysis and offer a summary estimate of effect to also provide the experts with some qualitative descriptor of our confidence in the repeatability of our meta-analysis estimate. And for that, we use the grade system that's endorsed by the Cochrane collaboration. In a nutshell, grade outlines 8 different categorical predictors, and depending on how each of those are assessed across the study data set, one arrives at an overall rating for that data set, varying from high to very low. And that, again, is an indication of our confidence in the repeatability of our meta-analysis estimate. It's in no way a reflection on the quality of those individual studies, for example. So the low rating there suggests that we think it's probable that the magnitude, at least, of that odds ratio would change potentially with future research. And by way of example, a later piece of research using a different outcome, measuring salmonella in subbiliac lymph nodes as opposed to fecal shedding, found a significant reduction in prevalence associated with probiotic feeding. And in contrast, there was a non-significant association reported in 2 cross-sectional studies. Feed management was investigated largely by observational studies, and so we were able to identify a number of predictors which were non-significant in observational, usually cross-sectional studies for culture of Salmonella on farm, including use of total ration versus non-mixed, giving the animals well water as opposed to chlorinated town water, and feeding dairy cattle from a bottle relative to a bucket. These were all non-significant predictors of culturing Salmonella on farm in cross-sectional studies. And in contrast, again, by inconsistent, I mean across different studies, the direction of the treatment effect changed. So the inconsistent predictors included withholding feed pre-slaughter, permitting forage grazing to dairy cattle, and feeding distillers' grains in observational studies, although you can see that was also studied in several controlled trials that consistently found a non-significant treatment effect. So vaccination was the most studied intervention category in our data set. And again, we were able to pool findings from several studies looking at sampling 2 different populations, one being adult dairy cattle and one being feedlot cattle, and in both cases, the treatment was vaccination with a variety of different Salmonella vaccines, and the outcome then was fecal shedding, pardon me. And you can see that they had similarly modest treatment effects in both of those populations. We also captured 3 other controlled trials which reported significantly increased odds of dam seroconversion, of colostral antibody production, and calf seroconversion after administration of a variety of different Salmonella vaccines. And in contrast, we captured 5 observational studies which reported non-significant association between vaccination and culture on farm. So we had our miscellaneous, or other category, and across 6 observational studies, the majority reported a significant association between increasing size of farm and increasing odds of Salmonella culture on farm. And to me, maybe the most interesting one in our other category was the recent observational study from the United States, which supports the hypothesis of vertical transmission of Salmonella from dam to offspring. So our study had a number of limitations. If any of you have been involved in systematic reviews, rapid systematic review is in fact a contradiction in terms usually, and so we had to streamline our methods somewhat to meet the timeline. For the same reason, we also had to restrict data extraction to English, French, or Spanish, and so we excluded 56 papers. We had a relatively small number of studies in our data set, which were similar enough that we could combine them for meta-analysis. And particularly in the meta-analysis data sets, prevalence was reported much more frequently than concentration as the outcome that was measured. And that to me is an important data gap, since identifying Salmonella interventions which are effective at reducing concentration can have a very major public health impact downstream. Finally, our objective was to summarize the publicly available evidence for control measures. That meant that we did not extract any data from proprietary sources, which probably had more effect in the processing data set, and also we didn't include anything from pathogenesis studies. So overall, in our data set, biosecurity, particularly external biosecurity or maintaining a closed herd, and vaccination were the broad intervention groups that were most consistently reported effective. That said, as a practitioner, I think a multiple hurdle approach would be more likely to be recommended by most veterinary practitioners, and that's tough to study in primary research. I think in the future, some mathematical modeling might be a useful way to tackle that problem. And finally, personally, I wonder if we will in the future be discussing Salmonella interventions specifically targeting protection of the bovine fetus. So with that, I'd like to thank my friend and colleague Janet Harris, who helped us get all the papers for this project. And improbably, there are 2 veterinary practitioners here at this conference in Dublin from the very small town of Vermilion, Alberta. And our part of Alberta is known as Big Sky Country. So at the risk of bragging, I wanted to end with a picture of our native land. And thank you very much for your attention.
Thank you very much, Barbara, for that very interesting talk. Have we any questions? Please.
Just regarding the vertical transmission from cow to dam, what did they test in the calf? What species did they isolate?
Would you mind repeating the question, please?
Sorry about that. Just in regarding the paper on the vertical transmission of Salmonella from dam to fetus,
There we go.
how did they sample the calf, and what Salmonella types did they culture?
Okay, I would encourage you to the papers by Hansen, and it's really interesting. They identified a farm which had clearly endemic Salmonella. They took a convenience sample of 20 cows, and so when the investigators were calving, or when the investigators were on farm and a cow was calving, then they sacrificed the calf immediately. Within 2 minutes of parturition, it was euthanized. They sampled, I think, 11 different tissues from stem to stern. So they sampled tonsil, variety of lymph nodes, cecal content, rectal content. Of those 20 calves, it seems to me 10 were positive on more than one culture, including stuff way downstream like cecal contents, lymph nodes. It's a really interesting piece of work.
Any other questions? Okay, Barbara, thank you very much indeed.